Hello and welcome. Today, I'm going to whisper to you about a fascinating topic, the discovery of penicillin. So, get comfortable, relax, and let's dive into this incredible story together. Before the discovery of penicillin, the world was a very different place when it came to medicine. Imagine a time when even a small cut or scratch could lead to a deadly infection. Bacterial infections were a significant cause of death, and doctors had limited means to combat them. Treatments were rudimentary, often involving harsh chemicals or methods that were more harmful than the infections themselves. In the early 20th century, the medical community was desperately searching for a solution. People were dying from infections that today seem easily treatable, like strep throat or a simple skin infection. Hospitals were filled with patients suffering from various bacterial infections, and there was a pervasive sense of helplessness among doctors. The common practice for treating infections involved the use of antiseptics and disinfectants, but these methods were not always effective and often caused more harm than good. Infections like pneumonia, tuberculosis, and sepsis were rampant, with no reliable cure. The mortality rates for such infections were alarmingly high, creating a pressing need for a breakthrough in medical science. To give you a better idea of the state of medicine at that time, let's look at some statistics. For example, pneumonia, which is now easily treatable, was once a leading cause of death. In the United States, during the early 1900s, pneumonia and influenza together were responsible for more deaths than any other cause. Similarly, tuberculosis was rampant, and its treatment was limited to rest and fresh air, with little hope for recovery. Medical practices were often crude and painful. Surgeons performed operations without the aid of modern anesthesia, and antiseptic practices were still in their infancy. The risk of post-surgical infections was extremely high, and many patients succumbed to infections after what would be considered routine surgeries today. The need for a reliable antibacterial treatment was evident, and the medical community was on the lookout for any potential solution. Alexander Fleming's early life. Now, let's talk about the man who changed everything, Alexander Fleming. Born on August 6, 1881, in Lockfield, Scotland, Fleming was the son of a farmer. Growing up in the rural countryside, he developed a keen sense of observation and a love for nature. These traits would later play a crucial role in his scientific career. Fleming was a bright student, and he moved to London to pursue his education. He studied at St. Mary's Hospital Medical School, where he excelled in his studies. Fleming's curiosity and dedication to science were evident from an early age. He joined the research department at St. Mary's after graduating, where he worked under the guidance of Sir Almroth Wright, a pioneer in vaccine therapy. During his early years at St. Mary's, Fleming conducted various experiments and developed a reputation as a meticulous researcher. His work on bacteriology and immunology laid the foundation for his future discoveries. Fleming was known for his keen observational skills, which played a crucial role in his discovery of penicillin. Fleming's early life was shaped by his upbringing in the Scottish countryside, where he developed a strong work ethic and a deep appreciation for the natural world. These early experiences influenced his approach to science, fostering a sense of curiosity and a desire to explore the unknown. His fascination with how the human body fought infections led him to pursue a career in medicine, where he could combine his love for science with his desire to help others. Fleming's journey to becoming a scientist was not straightforward. He initially worked in a shipping office before receiving a small inheritance that allowed him to attend medical school. His academic prowess soon became evident, and he won a scholarship to study at St. Mary's Hospital Medical School in London. There, he joined the research team led by Sir Almroth Wright, a prominent bacteriologist known for his work on vaccines and immunology. Under Wright's mentorship, Fleming honed his skills in bacteriology and developed a keen interest in studying how the immune system responded to infections. He was particularly intrigued by the body's natural defenses against bacteria and viruses. This curiosity would eventually lead him to his groundbreaking discovery of penicillin. The accidental discovery. The story of penicillin begins in 1928 with a rather serendipitous event. Fleming, who was working at St. Mary's Hospital in London, returned from a vacation to find that one of his petri dishes containing Staphylococcus bacteria had been contaminated by a mold. This mold, Penicillium notatum, had created a clear zone around itself where the bacteria had been killed. At first, Fleming didn't realize the significance of his discovery. However, his curiosity led him to investigate further. He identified the mold as a strain of Penicillium and observed that it produced a substance that could kill a wide range of bacteria. Fleming named this substance penicillin. Fleming's initial experiments with penicillin were rudimentary but promising. He found that penicillin was effective against many common bacteria that cause serious infections, including Streptococcus, Staphylococcus, and the bacteria that cause diphtheria and pneumonia. This discovery was groundbreaking, as it suggested a potential treatment for bacterial infections that were previously untreatable. The discovery was accidental, but it was Fleming's keen scientific mind that recognized its potential. He observed that the mold not only inhibited the growth of the bacteria, but also destroyed them. This clear zone around the mold, known as the zone of inhibition, indicated that the substance produced by the mold had antibacterial properties. Fleming conducted a series of experiments to test the effectiveness of penicillin. He found that it was capable of killing a wide range of harmful bacteria, while leaving human cells unharmed. This was a significant finding, as it suggested that penicillin could be used to treat bacterial infections without causing harm to the patient. Despite the promising results, Fleming faced significant challenges in developing penicillin into a usable form. The substance was unstable and difficult to produce in large quantities. Additionally, Fleming's work was met with skepticism by the scientific community. Many of his colleagues doubted the practical applications of penicillin, primarily because it was difficult to produce in large quantities and it degraded quickly. Fleming published his findings in 1929, but the response was lukewarm. The potential of penicillin remained largely unrecognized for several years. Fleming continued his research, but he lacked the resources and support needed to turn penicillin into a viable drug. It wasn't until a decade later that other scientists took up the challenge of developing penicillin into a practical treatment. Fleming's perseverance and belief in his discovery never wavered. He continued to conduct experiments and refine his methods, hoping that one day the significance of penicillin would be recognized. His determination paid off when Howard Florey and Ernst Boris Chain, researchers at the University of Oxford, took an interest in his work. Initial reactions and challenges. Despite the promising results, Fleming faced significant challenges in developing penicillin into a usable form. The substance was unstable and difficult to produce in large quantities. Additionally, Fleming's work was met with skepticism by the scientific community. Many of his colleagues doubted the practical applications of penicillin, primarily because it was difficult to produce in large quantities and it degraded quickly. Fleming published his findings in 1929, but the response was lukewarm. The potential of penicillin remained largely unrecognized for several years. Fleming continued his research, but he lacked the resources and support needed to turn penicillin into a viable drug. It wasn't until a decade later that other scientists took up the challenge of developing penicillin into a practical treatment. Fleming's perseverance and belief in his discovery never wavered. He continued to conduct experiments and refine his methods, hoping that one day the significance of penicillin would be recognized. His determination paid off when Howard Florey and Ernst Boris Chain, researchers at the University of Oxford, took an interest in his work. From discovery to development. Despite these obstacles, Fleming did not give up. His persistence eventually paid off when he published his findings in 1929. Unfortunately, the response was lukewarm, and the potential of penicillin remained largely unrecognized for several years. It wasn't until a decade later that other scientists took up the challenge of developing penicillin into a viable drug. Howard Florey and Ernst Boris Chain, researchers at the University of Oxford, were the ones who eventually succeeded in turning Fleming's discovery into a practical treatment. They recognized the importance of penicillin and worked tirelessly to purify and mass-produce it. Their efforts, combined with Fleming's initial discovery, laid the groundwork for one of the most significant medical advancements in history. Florey and Chain faced numerous challenges in their quest to produce penicillin on a large scale. The process was complex, and the yields were initially very low. They needed significant resources to continue their work, which led them to seek support from the US and British governments, especially as World War II intensified. The urgency of war and the need for effective treatments for wounded soldiers provided the impetus for the mass production of penicillin. The US government invested heavily in the project, recognizing the potential of penicillin to save countless lives on the battlefield. Pharmaceutical companies were mobilized to scale up production, and by 1944, penicillin was being produced in large quantities. Despite the challenges he faced, Fleming's discovery of penicillin laid the groundwork for a revolution in medicine. His initial findings, although met with skepticism, caught the attention of other researchers who saw the potential in penicillin. Among these were Howard Florey and Ernst Boris Chain, researchers at the University of Oxford, who would play crucial roles in turning penicillin from a laboratory curiosity into a life-saving drug. Howard Florey, an Australian pathologist, and Ernst Boris Chain, a German biochemist, recognized the immense potential of Fleming's work. They understood that if penicillin could be produced in sufficient quantities and purified effectively, it could revolutionize the treatment of bacterial infections. In 1939, they began their research into penicillin with a small team at the Sir William Dunn School of Pathology at Oxford. Florey and Chain faced numerous technical and logistical challenges. Penicillin was not only difficult to produce, but also unstable and prone to degradation. One of their first tasks was to develop a method to extract and purify penicillin in a form that could be used to treat patients. This process involved cultivating the penicillium mold and then extracting the penicillin it produced. Overcoming production challenges. The team's early attempts at producing penicillin yielded very small amounts, insufficient for treating patients. However, they persevered, refining their methods and scaling up production bit by bit. One of their key breakthroughs came when they discovered that corn steep liquor, a byproduct of corn processing, significantly increased the yield of penicillin from the mold cultures. Despite these improvements, the team still struggled to produce enough penicillin for clinical trials. They needed to find a more efficient way to produce the drug, which led them to seek help from industrial partners. This collaboration marked the beginning of a significant partnership between academia and industry, which would be crucial for the mass production of penicillin. The onset of World War II added urgency to their efforts. The need for effective treatments for wounded soldiers drove the British and American governments to invest heavily in the production of penicillin. In the United States, the government provided funding and resources to pharmaceutical companies to scale up production. Companies such as Pfizer and Merck played pivotal roles in developing large-scale fermentation processes to produce penicillin. Penicillin in World War II. The mass production of penicillin began in earnest during World War II. The drug proved to be a game-changer on the battlefield, saving countless lives by treating infected wounds and preventing the spread of infections. Penicillin's ability to effectively combat bacterial infections made it an essential part of the medical supplies for Allied forces. The first major test of penicillin's effectiveness came during the North African Campaign in 1943. The results were nothing short of miraculous. Soldiers who would have died from infections were able to recover and return to duty. The use of penicillin reduced the mortality rate from bacterial infections dramatically and helped to boost the morale of both the troops and the medical staff. One notable success story was the treatment of severe infections such as gangrene, which was common among soldiers with battlefield injuries. Before penicillin, these infections often led to amputations or death. With penicillin, many soldiers were able to avoid such drastic outcomes, preserving their limbs and their lives. Widespread acceptance and Nobel Prize. The success of penicillin during the war did not go unnoticed. Its effectiveness in treating bacterial infections led to widespread acceptance in the medical community. By the end of the war, penicillin was being hailed as a miracle drug, and its production was ramped up to meet the increasing demand from both military and civilian hospitals. In 1945, Alexander Fleming, Howard Florey, and Ernst Boris Chain were awarded the Nobel Prize in Physiology or Medicine for their contributions to the development of penicillin. This recognition was a testament to the impact of their work and the lives saved by their discovery. Penicillin had transformed medicine, turning once deadly infections into manageable conditions. The Nobel Prize highlighted the collaborative nature of scientific discovery. Fleming's initial observation, combined with Florey and Chain's determination and innovation, showcased how teamwork and perseverance could lead to breakthroughs that changed the world. Their work laid the foundation for the development of other antibiotics, marking the beginning of the antibiotic era. Post-war developments and impact. The end of World War II marked the beginning of a new era in medicine. The success of penicillin spurred further research into antibiotics, leading to the discovery of other life-saving drugs such as streptomycin, tetracycline, and erythromycin. These antibiotics broadened the range of bacterial infections that could be treated, saving millions of lives worldwide. The widespread availability of antibiotics also had a profound impact on public health. Diseases that were once common and deadly, such as pneumonia, tuberculosis, and syphilis, could now be effectively treated. This led to a dramatic decrease in mortality rates and an overall improvement in quality of life. Penicillin's success also influenced medical practices and standards. Hospitals adopted more stringent sterilization and hygiene protocols, reducing the incidence of hospital-acquired infections. The ability to treat infections more effectively also allowed for advancements in surgery and other medical procedures, as the risk of postoperative infections was greatly reduced. Transforming medicine. Penicillin's discovery marked a turning point in the history of medicine. Before its discovery, bacterial infections were often life-threatening, with limited treatment options. The introduction of penicillin transformed the treatment of these infections, saving countless lives and ushering in the antibiotic era. Penicillin became the prototype for the development of a wide range of antibiotics that followed. The impact of penicillin on the medical field was profound. It allowed for the effective treatment of bacterial infections that were previously fatal. Pneumonia, once a leading cause of death, could now be treated effectively. Syphilis and gonorrhea, sexually transmitted infections that cause significant morbidity, were now curable. Even common infections like strep throat and skin infections could be treated easily, reducing the burden on healthcare systems and improving overall public health. Penicillin's success also encouraged further research into antibiotics, leading to the discovery of other significant drugs such as streptomycin, which was effective against tuberculosis, and tetracycline, which had a broad spectrum of activity against various bacterial infections. These antibiotics expanded the arsenal available to doctors, allowing for more effective and targeted treatments. Penicillin's impact on public health. The introduction of penicillin had a dramatic effect on public health. Mortality rates from bacterial infections plummeted and life expectancy increased. Diseases that once decimated populations were now under control, leading to a significant improvement in quality of life. Penicillin's impact was particularly notable in the treatment of childhood diseases. Infections that commonly affected children, such as scarlet fever and rheumatic fever, became manageable, drastically reducing child mortality rates. The availability of effective antibiotics meant that children could recover quickly from infections, reducing the long-term health impacts and allowing them to lead healthier lives. The success of penicillin also led to the development of vaccination programs. With the ability to treat infections effectively, attention turned to prevention. Vaccines for diseases like diphtheria, tetanus, and whooping cough were developed and widely implemented, further reducing the incidence of these diseases and contributing to overall public health improvements. Modern antibiotic challenges. While penicillin and other antibiotics revolutionized medicine, they also brought new challenges. One of the most significant issues facing modern medicine is antibiotic resistance. Bacteria are remarkably adaptable, and over time, many strains have developed mechanisms to resist the effects of antibiotics. This has led to the emergence of superbugs that are difficult, if not impossible, to treat with existing drugs. The rise of antibiotic resistance has prompted a global health crisis. Infections that were once easily treatable have become more complex and dangerous. This has necessitated a renewed focus on the development of new antibiotics and alternative treatments. Researchers are exploring innovative approaches, such as bacteriophage therapy, which uses viruses to target and kill bacteria, and the development of drugs that disrupt bacterial communication systems, making them less virulent. Efforts to combat antibiotic resistance also involve promoting the responsible use of existing antibiotics. This includes guidelines for appropriate prescribing practices, reducing the use of antibiotics in agriculture, and educating the public about the dangers of overuse and misuse of these drugs. By preserving the effectiveness of current antibiotics and investing in new treatments, the medical community hopes to stay ahead of resistant bacteria. Penicillin today. In conclusion, today, penicillin remains an essential tool in the medical arsenal. While many newer antibiotics have been developed, penicillin and its derivatives are still widely used to treat a variety of infections. They are particularly effective against streptococcal infections, syphilis, and certain types of pneumonia. The legacy of penicillin is evident in the countless lives it has saved and the doors it has opened for further medical advancements. The discovery and development of penicillin is a story of curiosity, perseverance, and collaboration. From Fleming's accidental discovery to the tireless efforts of Florian Chain, it highlights the importance of scientific inquiry and the impact that one breakthrough can have on the world. Penicillin's legacy continues to this day, reminding us of the power of science and the endless possibilities it holds for improving human health. As we look to the future, the story of penicillin serves as an inspiration for continued innovation in medicine. The challenges we face today, such as antibiotic resistance, require the same spirit of determination and collaboration that led to the discovery of penicillin. By learning from the past, we can continue to advance medical science and improve the lives of people around the world. The impact of penicillin goes beyond its medical applications. It has become a symbol of the transformative power of scientific discovery. The journey from Fleming's laboratory to the widespread use of penicillin is a testament to the importance of research, collaboration, and persistence. It shows us that even the smallest observations, like mold growing on a petri dish, can lead to groundbreaking discoveries that change the course of history. Penicillin's story also underscores the importance of global collaboration in addressing health challenges. The production and distribution of penicillin during World War II required coordinated efforts between scientists, governments, and pharmaceutical companies across different countries. This collaboration set a precedent for how the global community can come together to tackle health crises, a lesson that remains relevant today. In conclusion, the discovery of penicillin is more than just a milestone in medical history. It is a story of hope, innovation, and the relentless pursuit of knowledge. It reminds us of the incredible potential of science to improve our lives and the importance of supporting research and innovation. As we continue to face new health challenges, the legacy of penicillin serves as a beacon, guiding us towards a future where we can overcome even the most formidable obstacles through the power of scientific discovery. Thank you for joining me on this journey through the discovery of penicillin. I hope you found it as fascinating as I do. Remember to stay curious, keep exploring, and never underestimate the power of a single idea to change the world. Until next time, take care and stay curious.